Mitochondria on the brink, Barth Syndrome's hidden attack on cellular power. Imagine your body's energy factories failing at the worst possible moment. That's exactly what happens in Barth Syndrome, a rare genetic disorder mostly striking boys. A glitch in the Tafazzin gene sabotages the production of cardiolipin, the lipid that keeps mitochondria running smoothly, while letting its toxic cousin, monolysocardiolipin, pile up and throw everything off balance. The result? Weakened muscles, recurring infections, and life-threatening heart issues that often cut childhood tragically short. But here's where it gets controversial. Researchers now think they have uncovered a new genetic lever, ABHD18, that could shift this grim outlook. Could targeting this obscure gene really outmaneuver the root defect in Tafazzin? In a recent international study published in Nature, genetic suppression features ABHD18 as a Barth Syndrome therapeutic target. Scientists from the Hospital for Sick Children, the University of Toronto, Scenic Biotech, and other institutions describe how blocking ABHD18 revives mitochondrial health in lab and animal models. Before this breakthrough, ABHD18 had no defined role. We only knew its name, not its function, recalls researcher Sana Massoud. By systematically screening for genes interacting with Tafazzin, Massoud and colleagues discovered that ABHD18 actively converts cardiolipin into monolysocardiolipin. Disabling ABHD18 tipped the balance back toward healthy lipid levels in cells and mouse tissues. And this is the part most people miss. Correcting Tafazzin itself remains a formidable task, so manipulating ABHD18 presents a more straightforward route. When the team administered a small molecule inhibitor, ABD646, MLCL levels plunged in preclinical models. Zebrafish with Barth-like symptoms regained stronger heartbeats, and patient-derived cells showed healthier mitochondrial structure and energy output. One of the most striking disease modifiers we have ever encountered, says Vincent Bloman of Scenic Biotech. Professor Jason Moffat, co-author and genetics expert at the University of Toronto, emphasizes that ABHD18 gives researchers a practical path forward toward therapies that address the disease's core imbalance. Yet this strategy raises provocative questions. Might inhibiting ABHD18 in healthy tissues cause unforeseen problems? Does focusing on gene suppression sideline promising gene replacement therapies for Tafazzin? Weigh in below. Do you agree that ABHD18 is the game-changer Barth Syndrome research has been waiting for? Or do you see risks that demand caution? We're eager to hear your perspectives.